Stem cells are among the most remarkable discoveries in modern biology because of their unique capacity for self-renewal and differentiation into specialized cell types. Unlike other cells in the body that are committed to a particular function, stem cells remain undifferentiated until they receive specific signals from their environment. This versatility allows them to become virtually any type of cell in the body, including neurons, astrocytes, and other glial cells that make up the intricate architecture of the brain. The ability to generate neurons is particularly significant because these cells are the fundamental units of communication within the nervous system, responsible for transmitting electrical and chemical signals that govern thought, behavior, and bodily function. When neural circuits are disrupted by conditions such as autism, epilepsy, or schizophrenia, the loss or dysfunction of specific neurons can severely impair communication across the brain, leading to a wide range of cognitive, emotional, and behavioral symptoms. The self-renewing property of stem cells means that they can divide repeatedly over long periods without losing their ability to differentiate. This feature is crucial for any therapeutic application because it provides a renewable source of cells that can be expanded in a laboratory setting before transplantation. By generating a sufficient number of healthy functional cells, researchers can potentially replace or repair neural networks that have been damaged or underdeveloped. Moreover, certain types of stem cells, such as mesenchymal stem cells, not only differentiate into neurons, but also release a variety of bioactive molecules that promote tissue repair, reduce inflammation, and support the survival of existing neurons. This dual function, cell replacement and neuroprotection, enhances the potential effectiveness of stem cell therapies in addressing complex neurological disorders that involve both structural and functional deficits in the brain. In addition to their regenerative potential, stem cells also offer a platform for understanding disease mechanisms at a cellular level. For disorders like autism, epilepsy, and schizophrenia, the exact causes often involve a combination of genetic, environmental, and developmental factors that disrupt neural connectivity. By studying stem cells derived from patients, scientists can observe how neurons and other brain cells develop, interact, and respond to various conditions. This approach provides insights into why certain circuits fail, why specific neurotransmitter systems become dysregulated, and how cellular interactions contribute to behavioral and cognitive symptoms. These insights are critical for designing therapies that do not simply treat symptoms, but target the root causes of dysfunction. Furthermore, the capacity of stem cells to integrate into existing neural networks is a critical aspect of their therapeutic potential. For a treatment to be effective, newly introduced neurons must not only survive, but also form functional connections with surrounding cells. Research has shown that under controlled conditions, stem cells can migrate to areas of damage, differentiate into appropriate cell types, and establish synaptic connections that restore some level of neural communication. This integration is particularly important in complex disorders where multiple circuits are involved, as it allows the therapy to support broader network functionality rather than addressing a single isolated defect. The combination of self-renewal, differentiation, neuroprotection, and integration makes stem cells a uniquely promising tool for repairing damaged neural circuits and potentially altering the course of neurological disorders in ways that conventional treatments cannot. Autism spectrum disorder is a complex neurodevelopmental condition characterized by challenges in social interaction, communication, and behavior, often accompanied by sensory sensitivities and repetitive patterns of activity. While the precise causes of autism remain multifactorial, research has highlighted that disruptions in neural connectivity, immune system function, and neuroinflammation play significant roles in the symptoms observed. Stem cell therapy has emerged as a promising approach because it addresses these underlying biological factors rather than focusing solely on symptom management. One of the central objectives of stem cell therapy in autism is to restore neural connectivity by generating new neurons and supporting cells that can integrate into existing circuits. Many studies suggest that individuals with autism may experience an imbalance in excitatory and inhibitory signaling in the brain, which can lead to hyper or hypoactive networks. Stem cells have the potential to correct these imbalances by differentiating into the appropriate cell types and establishing functional connections that enhance communication between brain regions. In addition to repairing neural circuits, certain types of stem cells, such as mesenchymal stem cells or induced pluripotent stem cells, have been shown to modulate the immune system. Emerging evidence indicates that immune dysregulation and chronic inflammation in the brain contribute to some of the cognitive and behavioral features of autism. Stem cells can secrete bioactive molecules that reduce inflammatory signals, promote tissue repair, and support the survival of existing neurons. This immunomodulatory effect helps create a healthier neural environment, which is essential for the proper functioning of developing circuits. By reducing inflammation, stem cells may also help mitigate oxidative stress and cellular damage, further supporting neural plasticity and cognitive flexibility. Neurogenesis, the process by which new neurons are formed, is another critical aspect of stem cell therapy in autism. Many neurological conditions, including autism, are associated with deficits in neurogenesis during development. By introducing stem cells capable of generating new neurons, researchers aim to replenish populations of cells that are underrepresented or functionally impaired. These new neurons can then participate in synaptic networks, enhancing information processing and communication across brain regions. The ability to promote neurogenesis is particularly important because it offers a mechanism for the brain to adapt and reorganize, potentially improving cognitive function and behavioral outcomes over time. Stem cell therapy may also support the production of neurotrophic factors, which are molecules that help neurons grow, survive, and form connections. These factors play a vital role in synaptic plasticity, which underlies learning, memory, and adaptive behavior. In individuals with autism, disruptions in synaptic plasticity may contribute to difficulties in social learning and flexibility of thought. By enhancing the availability of neurotrophic factors, stem cells can facilitate the strengthening and formation of synapses, creating a more robust and adaptable neural network. Beyond the biological mechanisms, stem cell therapy in autism is designed to work in a way that is personalized and targeted. Researchers are exploring methods to match stem cell types to the specific deficits present in individual patients, optimizing the potential for integration and functional recovery. Early clinical studies have reported improvements in social engagement, communication, and adaptive behaviors in some participants following stem cell treatment, suggesting that addressing the underlying cellular and neural dysfunction may translate into meaningful functional benefits. By focusing on neural connectivity, immune modulation, neurogenesis, and synaptic support, stem cell therapy represents a comprehensive strategy for addressing the complex biological factors that contribute to epilepsy. Is a neurological disorder characterized by recurrent, unprovoked seizures resulting from abnormal electrical activity in the brain. The underlying causes of epilepsy are diverse, ranging from genetic mutations to structural brain abnormalities and from neurochemical imbalances to prior brain injuries. Seizures occur when the delicate balance between excitatory and inhibitory signaling in neural circuits is disrupted, leading to excessive synchronized neuronal firing. Traditional treatments, including anti-epileptic medications and surgical interventions, often aim to control seizure frequency but do not repair the underlying neural dysfunction. Stem cell therapy presents a novel approach by targeting the cellular and network-level disruptions that contribute to epileptic activity, offering the potential to restore balance within the brain's circuitry. One of the primary mechanisms by which stem cells may help in epilepsy is through the generation of inhibitory neurons. In many forms of epilepsy, particularly those associated with cortical malformations or post-injury changes, the inhibitory neurons that normally suppress excessive firing are reduced or dysfunctional. Transplanted stem cells can differentiate into these inhibitory neurons, integrating into hyperactive circuits and providing the necessary restraint to reduce seizure activity. Animal studies have demonstrated that such integration can decrease hyperexcitability in affected regions, effectively reducing the frequency and severity of seizures. This targeted approach addresses the root cause of the abnormal electrical activity rather than merely masking the symptoms. Beyond cell replacement, stem cells also release neurotrophic factors and signaling molecules that support the health and survival of existing neurons. Neurotrophic factors such as brain-derived neurotrophic factor, BDNF, play critical roles in maintaining synaptic stability and promoting neural plasticity. In epileptic brains, neurons are often stressed or damaged due to repeated seizure activity, and these neurotrophic signals can help repair or protect them. By enhancing neuronal resilience, stem cells create a more stable neural environment, which reduces the likelihood of excessive synchronous firing that triggers seizures. Stem cells can also influence inflammation in the brain, which is increasingly recognized as a contributing factor in epilepsy. Chronic neuroinflammation can exacerbate excitability and impair normal neural network function. Certain stem cells, particularly mesenchymal stem cells, have immunomodulatory properties, releasing molecules that reduce inflammation and mitigate the toxic effects of inflammatory cytokines. This creates a more balanced environment in which neurons can function more normally, reducing both the frequency and severity of seizures over time. Another aspect of stem cell therapy in epilepsy is their potential to support synaptic plasticity and network reorganization. Epileptic circuits often become maladaptively rewired due to repeated seizure activity, leading to persistent abnormal connectivity. Stem cells may facilitate the formation of new functional synapses and the pruning of aberrant connections, helping to restore a healthier pattern of neuronal communication. This restructuring can improve the brain's ability to regulate electrical activity and maintain homeostasis, which is crucial for long-term seizure control. Delivery methods and timing also play an important role in the efficacy of stem cell therapy. Precise targeting of affected brain regions allows the transplanted cells to migrate, differentiate, and integrate effectively, maximizing their therapeutic potential. Early intervention may also enhance outcomes by limiting ongoing neuronal damage and providing support before maladaptive networks become entrenched. By combining these mechanisms, generation of inhibitory neurons, neuroprotection, immunomodulation, and network repair, stem cell therapy offers a comprehensive approach to addressing epilepsy at the cellular and circuit level. This represents a significant shift from symptom management to functional restoration, providing hope for improved seizure control and enhanced quality of life for individuals with this complex disorder. Schizophrenia is a complex psychiatric disorder that affects thought processes, perception, emotion, and behavior. It is associated with disruptions in multiple neurotransmitter systems, including dopamine and glutamate, as well as structural and functional abnormalities in neural circuits connecting key brain regions such as the prefrontal cortex, hippocampus, and striatum. Traditional treatments, including antipsychotic medications, often focus on controlling symptoms like hallucinations, delusions, and disorganized thinking, but do not repair the underlying cellular or network-level dysfunction. Stem cell therapy offers a novel approach by potentially restoring the integrity of these neural circuits, providing a foundation for long-term improvements in cognitive and behavioral function. One of the primary ways stem cells may benefit individuals with schizophrenia is through their ability to replace or repair dysfunctional neurons. Many studies have shown that specific neuronal populations, such as inhibitory interneurons, are reduced or impaired in schizophrenia, leading to imbalances between excitatory and inhibitory signaling. Stem cells can differentiate into these specific neuron types and integrate into existing circuits, helping to restore balance. By providing new functional cells, the brain is better able to regulate electrical activity, reduce dysregulated signaling, and support coherent communication between regions that are critical for cognitive and emotional processing. Beyond replacing lost or damaged cells, stem cells can also modulate synaptic plasticity, which is crucial for learning, memory, and adaptive behavior. In schizophrenia, synaptic connections are often weakened or improperly formed, leading to deficits in working memory, attention, and executive function. Stem cells release neurotrophic factors such as brain-derived neurotrophic factor, BDNF, and glial cell line-derived neurotrophic factor, GDNF, which enhance the survival, growth, and connectivity of neurons. These factors promote the strengthening of existing synapses and the formation of new functional connections, enabling the brain to reorganize itself and improve communication between critical networks. Neuroinflammation is another factor implicated in schizophrenia, with evidence suggesting that chronic low-grade inflammation can exacerbate neural dysfunction and contribute to symptom severity. Certain stem cells, particularly mesenchymal stem cells, possess immunomodulatory properties that reduce inflammatory signaling in the brain. By controlling inflammation, stem cells create a more supportive environment for neural repair and synaptic integration. This reduction in neuroinflammation also protects existing neurons from ongoing damage, which is essential for maintaining long-term functional gains. Stem cells may also help normalize neurotransmitter systems that are disrupted in schizophrenia. For instance, dopamine dysregulation in the prefrontal cortex and striatum contributes to both positive symptoms, like hallucinations, and negative symptoms, such as lack of motivation or emotional blunting. By integrating into affected circuits and supporting the health of surrounding neurons, stem cells can help restore more balanced dopamine signaling. Similarly, glutamatergic pathways, which are critical for synaptic plasticity and cognitive function, may benefit from stem cell-mediated network repair and support. The timing, type, and delivery method of stem cells are critical factors in achieving effective outcomes. Personalized approaches aim to match specific stem cell types to the deficits present in each patient and to target affected brain regions accurately. Experimental and preclinical studies have shown improvements in cognition, behavior, and synaptic function following stem cell transplantation, suggesting that these therapies can address the core neurobiological disruptions of schizophrenia. By repairing neuronal circuits, enhancing synaptic plasticity, modulating inflammation, and supporting neurotransmitter balance, stem cell therapy represents a multifaceted approach to a disorder that has historically been treated primarily through symptom management. While stem cell therapies offer significant potential for treating neurological and psychiatric disorders, it is important to recognize that this field remains experimental and extensive research is required to ensure safety, efficacy, and long-term benefits. One of the central challenges lies in controlling stem cell differentiation. Stem cells are inherently versatile, which is both a strength and a potential risk. Without precise regulation, stem cells may differentiate into inappropriate cell types, fail to integrate properly into neural circuits, or even form unwanted tissue growths. Researchers use a combination of biochemical signals, growth factors, and gene editing techniques to guide stem cells toward the desired lineage, but this process requires careful monitoring and validation to prevent unintended consequences. Another critical consideration is the integration of transplanted stem cells into existing neural networks. For stem cell therapy to be effective, the new cells must not only survive after transplantation, but also establish functional connections with surrounding neurons. Achieving this integration is particularly challenging in complex disorders like autism, epilepsy, and schizophrenia, where multiple circuits may be disrupted or maladaptively rewired. Researchers are exploring methods to enhance cell migration, axonal growth, and synaptic connectivity to improve the likelihood that transplanted cells will become fully functional participants in the brain's existing networks. Safety is a paramount concern in clinical applications. Stem cell therapies must be carefully screened to prevent immune rejection, tumor formation, or the introduction of genetic abnormalities. Immune compatibility is especially important when using allogeneic stem cells, cells derived from donors rather than the patient's own tissue, because the recipient's immune system may recognize them as foreign and mount an attack. Techniques such as autologous stem cell derivation, where cells are taken from the patient and reprogrammed, can reduce this risk, but they also present technical and logistical challenges. Long-term efficacy is another area that requires careful investigation. Neurological disorders often involve chronic or progressive changes in brain structure and function, and it is essential to determine whether stem cell therapy provides lasting benefits or if repeated treatments are necessary. Monitoring outcomes over extended periods is critical to understanding how transplanted cells survive, integrate, and influence neural networks as the brain continues to develop or age. Additionally, researchers need to study potential interactions with existing medications or therapies to ensure that stem cell interventions do not produce unintended effects. Clinical trials are ongoing to test stem cell therapies for various neurological and psychiatric conditions, and early results are promising but preliminary. Improvements in cognition, behavior, and seizure frequency have been observed in some studies, but these outcomes must be validated in larger, controlled trials. Regulatory oversight is essential to ensure that treatments meet rigorous standards for safety, reproducibility, and ethical practice. Researchers are also developing standardized protocols for cell preparation, transplantation, and post-treatment monitoring to reduce variability and optimize outcomes. Advancements in complementary technologies such as gene editing, biomaterials for scaffolding, and imaging techniques are enhancing the precision and effectiveness of stem cell therapies. Gene editing can correct underlying mutations in patient-derived cells before transplantation, while scaffolds and supportive matrices can guide cell placement and improve survival. High-resolution imaging allows researchers to track transplanted cells in real time, assess integration, and adjust treatment strategies accordingly. Overall, while stem cell therapies hold extraordinary promise, their application requires careful consideration of differentiation control, integration, immune compatibility, long-term efficacy, and rigorous clinical validation. By addressing these challenges systematically, the field can move towards safe, effective, and reliable treatments that go beyond symptom management to fundamentally restore or repair neural function in complex disorders. Stem cell research represents a paradigm shift in treating neurological and psychiatric disorders because it focuses on repairing and regenerating brain function rather than simply managing symptoms. Traditional therapies, including medications and behavioral interventions, often target the outward manifestations of conditions like autism, epilepsy, and schizophrenia, but do not address the underlying cellular or circuit-level dysfunctions. Stem cells offer the potential to fundamentally alter the course of these disorders by restoring damaged neural networks, supporting synaptic plasticity, and promoting overall brain health. This regenerative approach aligns with the brain's inherent capacity for neuroplasticity, the ability to reorganize and adapt its circuitry in response to experience and environmental input, which is a crucial factor in long-term recovery. One of the key advantages of stem cell therapy is its multifaceted mechanism of action. Unlike conventional treatments that often target a single neurotransmitter or symptom, stem cells can simultaneously address multiple aspects of neural dysfunction. They can differentiate into neurons and glial cells, which are essential for maintaining the structural and functional integrity of neural networks. In addition, stem cells release neurotrophic factors that promote neuronal survival, growth, and connectivity, while also modulating inflammation and creating a supportive microenvironment for the brain to heal. By simultaneously repairing structural deficits, supporting cellular health, and reducing pathological inflammation, stem cells provide a comprehensive strategy for restoring neural function. The regenerative potential of stem cells is particularly valuable in disorders where neural connectivity is disrupted. In autism, for instance, imbalances in communication between brain regions contribute to social, cognitive, and behavioral challenges. Stem cells may help restore these connections, promoting more efficient signaling and functional coordination across networks. In epilepsy, stem cells can replace or support inhibitory neurons to reduce hyperexcitability and stabilize electrical activity in the brain. In schizophrenia, stem cells may repair damaged circuits, enhance synaptic plasticity, and normalize neurotransmitter signaling, thereby improving cognitive and behavioral outcomes. In each case, stem cells address the root biological mechanisms rather than only mitigating external symptoms. Another important aspect of stem cell therapy is its adaptability and potential for personalized medicine. Stem cells can be derived from the patient's own tissue, reprogrammed to correct genetic abnormalities, or engineered to target specific deficits in neural circuits. This personalized approach maximizes the likelihood of successful integration and minimizes risks such as immune rejection. Furthermore, advances in delivery methods, including precise targeting of affected brain regions and the use of supportive scaffolds, enhance the ability of stem cells to survive, differentiate, and integrate into existing networks. These technological refinements increase the potential for meaningful functional improvements. The long-term implications of stem cell therapy extend beyond immediate symptom reduction. By restoring neural circuits and supporting adaptive plasticity, stem cells can enhance the brain's capacity to learn, adapt, and respond to future challenges. Patients may experience improvements not only in core symptoms, but also in cognitive flexibility, social engagement, and overall quality of life. This approach fosters a more sustainable form of recovery because it strengthens the underlying architecture of the brain rather than providing temporary relief. Overall, stem cell research embodies a fundamental shift in how neurological and psychiatric disorders can be approached. By leveraging the brain's inherent regenerative capacity, focusing on circuit repair and synaptic support, and providing tools for personalized intervention, stem cell therapies offer the potential to move beyond symptom management toward meaningful, long-term restoration of brain function. This paradigm emphasizes recovery at the cellular and network levels, enabling patients to experience improvements in cognition, behavior, and overall life quality in ways that traditional treatments alone cannot achieve. When these regenerative strategies are applied effectively, they have the potential to transform the treatment landscape for neurological and psychiatric disorders. By addressing the underlying cellular and circuit-level dysfunctions, stem cell therapies move beyond temporary symptom relief and instead support lasting improvements in brain function. This approach harnesses the brain's natural ability to adapt and reorganize, allowing patients to regain cognitive, emotional, and behavioral capacities that were previously impaired. Ultimately, stem cell therapy represents a shift toward true restoration, offering hope for enhanced quality of life and long-term recovery in conditions that have historically been considered difficult to treat.